My pleasure. All right, everyone, you get to see it again, the crass capitalism. Please pay and give me a quarter. But this is what I wanted to show you, the opportunities of finding things like the rare Arctostaphylus franciscana, where they were reorienting the roads just before the Golden Gate Bridge. It did get moved. It took a gigantic crane, a gigantic truck, because when you have that much soil with water in it, it's very heavy. And it was moved in the middle of the night during a winter storm. There's its new location. It's in the Presidio, but I can't tell you where it is because it's secret. I'd have to kill you. That's what they tell me. So here we are in the High Sierra, Archeocephalus nevadensis, the Pine Mount Manzanita. Very widespread as you get up in higher and higher elevations. The fruit, they look like little apples. And since you've heard this all before, I'll just go through the slides a little bit. Arctocephalus patula, the greenleaf manzanita. Really lovely plant. And at lower elevations, the white-leaf manzanita, Arctostaphylos viscida. And again, now you can see the glossy inflorescence with the viscid glands on it that if you pinch, it'll stick to you. And over on the lower left, the Eldorado manzanita that looks like a little tiny Arctostaphylus canescens, the hoary leaf manzanita. And it's at a soil interface here with viscida, the white leaf manzanita on the right side. Other soil conditions create other opportunities, and here we are in outcroppings of oxasols, which are old tropical soils which accumulated in the Central Valley at the bottom of the foothills. And that's where we have an endemic Arctostaphylus myrtifolia, the Ione manzanita. And it's mixed in here with Arctostaphylus viscida still, the white leaf manzanita. It's a very odd plant, the Ione manzanita. It has red, smooth bark, but it has a blush of white over it. Very small plant otherwise, on very, very acidic soils. In between the greenleaf manzanita and the white-leaf manzanita is Arctostaphylus Miwaka, the Indian manzanita. So that is throughout the Sierra Nevada at the mid-ranges. It's been believed that this is of hybrid origin between the greenleaf and the white-leaf manzanita. It does have a burrow like the greenleaf manzanita, and it has white leaves like the white-leaf manzanita. The fruit, though, are quite different from both of them. And I did have a student who did some genetic studies, and we can't find that direct connection yet. In fact, it makes you believe that Arctostaphylus is as messy a group as possible. Here's that Hoary manzanita, Arctostaphylus canescens. It's a hairy white-leaved plant, and it's a dominant of chaparral in the coast ranges from Oregon south to the northern part of Monterey County. It's a really lovely plant, though. Little racemes with these large leaf-like bracts covered in hair. On Mount Tamalpais, the flowers have a pink blush to them, and they're usually white in most other places. And it's intermixed with Arctostaphylus glandulosa. So the bright green one that you see is glandulosa. The gray plant in the foreground is the Arctostaphylus canescens with the fog coming over in the upper left. Another common plant of the northern coast ranges is Arctostaphylus stanfordiana, Stanford's manzanita. This plant is really cool because it has no hair of any sort. It's very shiny green leaves. But the inflorescence is what's really interesting. The flowers will come out quite pink, but if you look at them when they're immature like this, the buds are these round little balls with very small bracts that encase them, and they're quite separate from in each other. And that's really unlike most other species in Arctostaphylos. Then you end up with a lot of other unusual ones that are often on different soils, like a variety of Arctostaphylos viscida called subspecies procella, which has different kinds of glandular hairs, and it's only found on serpentines in the North Coast ranges. Arctostaphylos bakeri, pretty much restricted to Sonoma County. It's a tall plant, a small tree, only found on serpentines in Sonoma County. And then there's an unusual subspecies of bakeri called the Cedars-Manzanita. And it's found only in about 3 places, one of which is the Cedars, a gigantic outcropping of serpentine that if you ever have the chance to go there, you should definitely do it. A botanist owns about 1/3 of it. You get to Marin County, and the serpentine endemic now is Arctostaphylos montana, the Mount Tamalpais manzanita, but it's kind of a short mounding plant, and it's restricted to serpentines in Marin County. It does have a subspecies, Arctostaphylos montana, subspecies Ravenii, which was found by Peter Raven when he was a 14-year-old who was living in San Francisco at the time. So it got named the Raven's Manzanita later. There's only one original individual left of this plant, although there are some cuttings and possibly some progeny from some plants at the UC Botanic Garden. I can't tell you where this one is either. It's also supposed to be secret. You go further south and follow the serpentines, you not only have Sargent cypress, but you pick up Obispoensis, Arctostaphylos obispoensis, the bishop's manzanita. That's mostly in the Santa Lucias and mostly in San Luis Obispo County, but there are some serpentine outcrops in southern Monterey County. This is another really lovely tall plant with gray leaves. Kind of reminds you a little bit of Arctostaphylos canescens, but not quite. It co-occurs with a lot of rare herbaceous species, especially Calochortus species, with a beautiful view of Morro Bay. Another widespread species from Central California to Baja is Archeocephalus Glauco, a species you probably know is big berry manzanita. We have forests of it in Northern California, just below San Jose. Really large fruit, the largest in the genus. Classic flowers, which are all kind of similar. So they don't really help you identify things except rarely. My first encounter with Archeocephalus Glauco was at Henry Coe State Park, and they were only tree size there, it seemed like. If you look at the lower right hand, that truck on the side is a gigantic 4-wheel truck. That's not a small truck. That's the kind that you have to climb into. And that means that Archeocephalus Glauco is about 30 to 40 feet tall. A huge, huge plant. Most of the species turn out to be along the coast, especially the central coast. And they are found where there is maritime climates with fog. Even in San Diego, you have maritime climates, but it's pretty close to the coast. It's a little more extensive as you go further north. And there's different kinds of maritime climates. If you're up high enough where the fog sits, you have a lot of fog drip. But if you're low and close to the ocean, you don't get that fog drip, but you do get the cover and the cooler temperatures. And that helps these plants conserve moisture during the dry summer. And a lot of plants have proliferated in that kind of area. So Bolinas Ridge is in Marin County, but Maritime climates go all the way to British Columbia, and there's a manzanita that follows them all the way. Here we've got a site with two different species, Archeocephalus glandulosa, subspecies glandulosa, which is really widespread, but then a Central Californian endemic, Arctostaphylos sensitiva, coin leaf manzanita. That plant is a little unusual because it has flowers that are four-merous, where the rest of the genus is basically five-merous, except for a sister taxa of sensitiva. And here's that sister taxa, Arctostaphylus numularia from Mendocino. Can't quite tell that it's only got four sepals and four bumps on the bottom of those corollas. In some sandy outcrops near Bonnie Doon in the Santa Cruz Mountains, we have another endemic, Arctostaphylus sylvicola, the Bonnie Doon manzanita, another white-leaved plant that looks pretty similar to Arctostaphylus canescens, but it does differ in a number of characters. Not too far away, above Big Basin, you can find two other local plants, Arctostaphylus sensitiva again, and an auriculate-leaved plant that's quite large, Arctostaphylus andersonii. That's the one in the foreground with the little pink flowers that are showing. Near Berkeley and Oakland, Arctostaphylus pallida, the Alameda Manzanita. This is new growth, so you're not going to find it looking like this except when it just begins to grow. A really lovely plant. Trapped by development on all sides. It's kind of a tough place. It needs fire to reproduce. How are we going to do that? Who knows? Another plant with auriculate leaves that's kind of unusual is Arctostaphylus imbricata on San Bruno Mountain. That's the northern part of San Mateo County near San Francisco. And if you walk to the right kind of place, you can get a view of San Francisco while you're standing in the middle of all those plants. This plant is really prostrate. I'll let you take a look at the city. The 49ers just beat the Rams last night, and I hope you're not Rams fans. Nearby is Arctostaphos Monterences. This is supposed to be a sibling species to that prostrate imbricata, but Monterences can be a gigantic plant. Otherwise, it's pretty similar. A little further south in Santa Cruz is Arctostaphylus glutinosa. This one has lots of short canescent hair and some glandular hairs associated with the inflorescence. Really very, very different. A little further south in the Pajaro Hills is Arctostaphylus pajaroensis. This picture is from Prunedale in a little county park called Manzanita Park. You're probably familiar with the horticultural selection where the new growth comes out bright red. It actually was selected from Manzanita Park. This is supposed to be a raceme for most of these plants, but if you look carefully, you can see that sometimes there's a second branch on these plants for those inflorescences. Here's a view of what Andersonii normally looks like, about 2.5 people tall, in full bloom. It's a really lovely plant with compact inflorescence of many branches. A little further inland is another auriculate leaf plant, Arctostaphylus gabalensis in the Gabalon Mountains, and it's only on granite outcroppings, which are pretty rare in the Gabalons. This granite is associated with the transverse ranges from Southern California, moved up through time. I'll show you close-up pictures of this plant later, but this is the habitat that it grows in. There is another manzanita associated with it, and this is at the very edge of maritime conditions. A lot of unusual soils also have endemics. So in Santa Cruz County in the Santa Cruz Mountains is one called Archeocephalus glutinosa. You've seen a close-up of it, but here's the stem, and you can see the Monterey shales that are the main soil that it's restricted to. This Monterey shale is very unusual. I took some children when my kids were young to Año Nuevo to go see the elephant seals, and the woman who was leading us out there stopped at one spot because it was me and 6 kids. And she picked up a little piece of the Monterey shale and said, if you hold this on your tongue, it'll stick. And all of the kids did that. And I had to take pictures of all these kids with rocks hanging out of their mouths, stuck to their tongues. So it's a very unusual soil condition, shall we say. I can't see you, so I can't tell if any of these jokes are working. So I hope it works. On sand dunes, really close to the coast, you can pick up Arctostaphylus tomentosa, subspecies tomentosa, and Arctostaphylos pumila, the sand mat manzanita. So here we're on stabilized sand dunes. We're only about a mile from the ocean. Down in San Luis Obispo County, really close to the nuclear power plant, is Arctostaphylos pechoensis in the Pecho Mountains. Very shaly soils in this area. If you've never been to that nuclear power plant, it's like going into a James Bond film when you first come around the corner and see it. It's very strange. Lots and lots of security. And then the widespread species in Southern California, Arctostaphylos pungens, which you must be familiar with, the really curly bracts. Now this is unusually curly. They do usually curl back a little bit. But of course, you always have to show the exceptions. Gabrielensis, the big round fruit with the seeds all fused together. And a resprouting, Arctocephalus glandulosa. This one, subspecies mollis, with the stiff hairs that are long, but it's a very white version of it. And this version has an older name, Glaucomollis, because it is so white. Another one in San Diego County, Glandulosa subspecies leucophila, also kind of white-leaved. And a Southern California endemic, Arctostaphylos rainbowensis, with these really lovely inflorescences. This one is another resprouter, but this one happens to be a diploid. So that's kind of a tour of a bunch of plants, but I thought I would tell you, how did I get involved in this group? Because this is not a group I wanted to work with. When I was a graduate student at UC Santa Barbara, we would be taken into the hills to be introduced to manzanitas. I remember being shown Arctocephalus refugioensis and glandulosa on the same field trip. And as soon as I turned around, I couldn't remember how to identify either of them. And it was always Arctostaphylos spa from that point on. So somehow I got pulled into this group. So that was when I got my job at San Francisco State, and I had a new graduate student who wanted to work with how do plants respond to fire that differ in their life histories. And Ceanothus and Arctostaphylos both have plants that resprout after fire and plants that are obligate cedars where the adults are killed by fire. Okay. Some will survive and some won't. The trick was that in the San Francisco area, there are no resprouting Ceanothus. So it would be a really long field trip for my graduate student to be able to work with Ceanothus species. So we decided we would work with Arctostaphylos, but it meant we had to learn how to identify them. And we spent about a year trying to figure out good places and how to identify the species. And ironically, at the end of that year, there was a workshop on manzanitas at UC Santa Barbara. And we went there, and there was a room full of cuttings of manzanitas from all over the place. And we walked around and we recognized them all. And we realized that that 1 year spent really intensely trying to identify them, we were suddenly in the top 5 of people globally who could identify manzanitas. It was hilarious. So we sort of got into that. But these 2 life histories have different selective histories as well. There's different kinds of pressure on them. They have plants on the left hardly ever die after a fire, but plants on the right, the entire population is killed. And both of these groups recover with seed that is stimulated by fire. And that fire is required for germination. The seeds remain dormant otherwise. So the resprouting plants are able to maintain most of their population and then add a few individuals with each fire. But the obligate cedars, the ones that are killed by fire, are completely dependent upon that stored seed in the soil to recover their populations. And that became the research project that we worked on. We didn't find super large differences in seed banks, but we did find really large differences in ploidy levels, which I'll talk about later, and in seed predation rates. But then another thing happened that also sucked me into this group. So if you look at this, that's a little cluster of baby manzanitas coming up. This happens to be after the car fire just a few years ago, but I'd been seeing this for a long time. These are seedlings coming from rodent caches that have all been buried together, that hopefully the rodents would come back to and consume later. Well, hopefully for the rodents. And they're all over the place. So this got me interested in the mutualism between a seed predator, rodents, and the plants themselves, Arctostaphylos. Arctostaphylos is the only plant with close relatives that has persistent soil seed banks where the seeds require fire for germination. So this really got me interested. Because these guys, my whole life, I had thought of as horrible, horrible things. They come and eat your plants, they eat your seeds. I could see nothing good about them, except the dichotomies are cute. But then I started going to post-fire stands, and I found out that when there's very high intensity, so if you look at this graph on the bottom, the further you are to the right, the higher the intensity of fire. What that means on the left side, if you follow the y-axis, the percentage of seedlings that are coming out of those rodent caches increases with the intensity of fire. So the rodent caches are really important for these plants to be able to recover after a high intensity fire. All of a sudden, I had to start rethinking things. But we had to prove that rodents were actually doing this. So do they actually bury seeds to sufficient depths? So we went out and we did a bunch of experiments in a few places. We took fruit and put them in the middle of a pizza pan with a lot of fluorescent powder so that the rodents could go get the fruit, but they'd have to walk over the fluorescent powder. And then we could follow their trails, and hopefully they would bury some of it, and we could dig up those caches and see how many fruit were there and how deep they buried it and things like that. It was really a fun project. As you might realize, it's not really easy to follow fluorescent powder trails in chaparral, but it can be done. And we found some interesting results. So here's sort of a diagram of what we found. So you've got a chaparral stand, and it grows to the soil. The surface of the soil is indicated there. And then there's a soil profile, shallow to deep. And the seeds have to be buried in the soil. But when you have a fire, the shallow parts of the soil are basically a kill zone. These seeds do not tolerate more than 125 Celsius. So they can't really survive a super high intensity fire if they're close to the soil surface. So they have to be buried a few centimeters deeper than that. So we found all these caches, and we determined that while there are a lot of seeds still in the shallow area, the rodent caches are mostly deep enough to survive even a very high intensity fire. So that was really cool. So these guys aren't so bad after all. And that was really a nice thing that made me rethink how I think about rodents. You always say things like, well, the ecosystem is a system and everything's tied together. But in reality, you have your biases about things. But this is one of those circumstances where it made me realize that plants, because they can't move, and because everybody wants to eat them, they are able to be selected to manipulate everything in their environment. And what manzanitas have done, have manipulated all of their seed predators to bury their seeds so that they can survive fires and the plants can recover after fires. So these guys aren't so bad, even though they consume almost all of the seed every year. So thank you, rodents. Of course, I also have to thank the rattlesnakes and the coyotes that eat these guys so that they don't recover all of their caches. Yeah, Ceanothus, or Arctostaphylos, is the center, or California is the center of Manzanita diversity. The green areas you see are mostly one species, but sometimes up to 3. And the peak of diversity is in the Central Coast ranges, although the North Coast ranges and the transverse ranges and northern Baja to San Diego are also very high species diversity for manzanitas. So that leads us to a question about how did these plants, evolve? These are not new plants. The fossil history of this group goes back over 15 million years. That's a long time. Humans are only a little over 1 million years in terms of our fossil record. So how did they get there? Now, 15 million years ago, there wasn't much of California. And almost all of these fossils that are old are found in Nevada. And at that time, Nevada was not the way you see it now, but it was a very high plateau. So it would have been cold. It would have been dry because the middle Miocene was a very warm time period. And eventually that high plateau collapsed into what we think of now as the basin and range. And eventually California became much more terrestrial. But at this point, there were 2 subduction zones throughout the state, and the Central Valley was mostly that sea that you see, really different circumstances. So let's back up a little bit and think about manzanitas from the perspective of where they came from. Who are their close relatives? And they're in a real small subfamily of the blueberry rhododendron family, the Ericaceae. And that subfamily is called the Arbutoideae, and it's named after madrone, the genus Arbutus. Okay. Now, what I want you to notice is that it's an early diverging lineage, and it doesn't seem to have any close relatives in the family. The closest relatives seem to be monotropes, like pine drops or snow plant. Madrones are forest trees, whereas all the other genera, except for Arctos, are shrubs in Mediterranean climates. Okay? Something you're familiar with. So let's talk about the Arbutoideae for a minute to give us a context for understanding the evolution of Arctostaphylos. So Arbutus, the madrones, there are about 10 species, and they're found in the Mediterranean area and in California and Mexico. And then we have Chromosomes, the summer holly. We've got 1 species in Southern California and a lot over in Mexico. And then we have 2 kind of local endemics in the southern part of the California floristic province, Ornithostaphylos, the Baja birdbush, and Xylococcus, the mission manzanita. And then you come to Arctostaphylos, the manzanitas. Now you've got 67 species with about 39 other subspecies, so well over 100 taxa, very speciose compared to all these other genera. And then you have this very unusual group, Arctos, the Alpine bearberries. And we won't talk about them too much because they're really very different, although they're probably closely related to Arctostaphylos. So let's take a look and see what these guys look like. Arbutus, fleshy fruit. You might be familiar with Arbutus unido from the Mediterranean because it's planted all over California. But our native, Arbutus menziesii, also has fleshy fruit and a nice orangey red bark. They have a very extensive fossil history, and they go back well over 30 million years. And there are some fossils that are over 50 million years, but there's some contention, so it's not quite clear. Nonetheless, it's a very old group. And they're distributed either around the Mediterranean, where there are 3 taxa, or in Western North America and Mexico. The summer holly, Chromosomes, our local one is diversifolia, but there are a lot of species in Mexico, parts of Central America. Again, fleshy fruits, very Western distribution. Ornithostaphylos, the Baja birdbush. We now have a very different kind of fruit. They're dry, but the flowers look familiar. Smooth, whitish bark is, if you know the whole group, that's not an unfamiliar look. And Xylococcus, the mission manzanita, also with dry fruit when it's mature. Their distributions are the southern end of the California floristic province. And finally, Arctostaphylos, a really, really diverse group with a huge distribution. But almost everything you're looking at is a single species, Arctostaphylos uva-ursi bearberry, which is mostly found in mesic or cold locations. Western North America has a few others, but that's where everybody is actually found, in the California floristic province. And finally, the really odd genus, Arctos, the Alpine bearberry. It does not have evergreen leaves. These leaves come out each year and then they die back. There are 2 species, Arctos alpina is worldwide in the alpine and boreal areas. The Arctos rubra is a North American taxa. They have succulent fruit, but they have really different seeds compared to the other species with succulent fruit. Very odd distribution because they're not at all adapted to Mediterranean climates. So let's take a look at the history of this group. And what I want you to see is we have 2 lineages of Arbutus. This is from an early study about 20 years ago. Arbutus in the top group that are colored blue are all the species from Europe and Africa. And that bottom group are all the species of Arbutus from North America and Mexico. And that's nice, at least the geography is fitting. But if you look where that arrow is, you see there's something odd. Those Arbutus are not clustered together, but instead the European African group are sister to all the other North American genera. So that's very unusual. That means that before that, well, at least it suggests that this group started in Western North America and moved eastward. And some of them evolved into different genera and others just kept being Arbutus and made it all the way to Europe. And that would have to be at least 28 to 35 million years ago to make it to Europe. Because the Atlantic Ocean became much too wide after that time period. for there to be much dispersal. That's a really old break between these two groups. So it's a very old lineage, this subfamily of the Ericaceae, 6 genera, mostly Western North American, different life forms, very, very different distributions and other characters, but only one of those lineages is really more diverse, and that's Arctostaphylos, the Manzanitas. Now, a lot of that diversity is probably due to the ecological shifts from woodlands of the Arbutus that are relatively moist with deeper soils, where the typical fire is a surface fire, and you have really very different animal communities of forest birds and rodents. Shrublands are mostly much drier with shallow soils. You only have canopy fires in those systems, and you have a different set of birds and rodents that live in those kinds of habitats. So that means that natural selection is going to be different. So let's just take a look at a limited number of characters, and we'll look at just reproductive characters. In the left panel, you can see Arbutus manzanitae, our local species, and it's a fleshy fruit. It has a number of locules or chambers, and each chamber has a whole bunch of seed, usually about 7 or 8. And I've dissected out one of those chambers, and you see the seeds there in the lower left-hand area. They have very thin endocarps, and that's the inside part of the fruit that protect the seed. Very small. And then Chromosomes next door on the right-hand side also has fleshy fruit, but very different kinds of seeds. Now the endocarps that enclose the seeds are stony and quite thick. And not only that, they've all fused together into a single structure. So if you've ever collected Chromosomes and broken them apart, you'll know that the seeds are all fused together like that with a very thick endocarps. If you're wondering what an endocarp is, just think of eating a cherry, and that pit in the middle is the thickened stony endocarp. And if you break that pit apart, you'll see the seed on the inside. So it's a very similar thing in the Arbutoideae, except that it's just much smaller than a cherry or a peach or something. If you go to some of the other genera, we've suddenly switched to dry fruit. So Xylococcus on the left-hand side there, very nice dry fruit, very stiff, dry mesocarp that's really difficult to remove from the endocarps. But notice they're also stony and thick, and they all fuse together. And then on the right-hand side, the big berry manzanita, again, dry fruit, very stony endocarps, and the seeds are all fused together. So something's going on because we've gone from Arbutus with very thin endocarps, where the seeds are all free, to all the other genera having very stony, thick endocarps protecting the seed, and they're all fused together. But of course, in Arctostaphylos, it goes crazy because they're quite variable in its fruit. So the left-hand panel just shows you 5 different species: Glauca, Canescens, Montana, Purissima, and Refugioensis. That's just a good range of sizes and types of seed. And if you look at the middle panel, the one labeled B, all the fruit have been removed, and you're just seeing the endocarps. So Glauca, all the seeds are fused together. The lower right-hand corner, Refugioensis, all the seeds are fused together. Canescens in the middle, the seeds are, in that particular case, were all free. Montana, there's a little bit of fusion, but some freeness. And Purissima in the lower left-hand corner, in that case, all the seeds happen to be free. Now, I keep using these squishy words like happen to be and sort of, and in this case. So if you look on the right-hand panel for just one of the species, Arctostaphylus montana, you'll see what I'm talking about. So I opened up 5 different fruit, and that's the amount of fusion that you see in those 5 different fruits. So in one case, number 1, all of the seeds were fused together, just like you were seeing in the other genera. In 2 and 3, there's partial fusion, but some freeness. And in 4 and 5, it's a little freer, but again, there's some partial fusion. So quite a lot of variability even within a single species, just different fruit. And that's going to be an important character to keep in mind, because you know that rodents are important in creating the seed banks for the species. So let's sort of summarize what we've been talking about here. If you look at the column called fruit, Arbutus and Chromosomes have fleshy fruit, but the other genera that we're focusing on here from Mediterranean climates have dry fruit. Now, the madrone and the summer holly also flower later in the spring and mature fruit late summer and into the fall. The others with the dry fruit, they flower much earlier. Arctostaphylos flowers in December, January, February, as do Xylococcus and Archeocephalus. And they mature fruit in late spring to very early summer. And you can imagine why that might be, because they're in a very dry habitat, and having fleshy fruit means you're pumping water into a structure during a dry season. So it's kind of an interesting shift there. The endocarps in the second column, they're thin in Arbutus, but they're stony and thickened in all the other genera. You go to the second column of endocarps, the Arbutus, madrone endocarps are all separate. And then all the others are fused that we were seeing in those pictures, except when you get to Arctostaphylos. Some of them are fused, like in Big Berry Manzanita, and some of them separate to some extent. The seeds in that column, transient just means that the seeds lose viability if they don't germinate roughly within a year. So they're not dormant seeds of any long-term nature. But you get to Arctostaphylos and it's completely different. They're dormant seeds. They need fire to stimulate them. And as a consequence, Arctostaphylos produces persistent, very long-term seed banks. You get to the fire column, everybody can resprout to some extent, except for in Arctostaphylos, where some species resprout, but about 2/3 of the species are obligate seeders. That might surprise you, because obligate seeding is actually an adaptation to fire. It permits the populations to respond to shifts in climate, to shifts in soils. But it seems kind of odd that you give up your population rather than being conservative and being able to resprout. So we can summarize this in a different way. If you look at the graph on the right, that's the family lineage. And ancestrally, succulent fruit and resprouting, the fleshy fruit, were probably there already. But by the time you get to Chromosomes, everybody past that point has thickened endocarps, and they're fused together. A little bit further down, all of them suddenly are dry fruit, and the fruit mature much earlier, and they flower much earlier. But only in Arctostaphylos do you have that seed dormancy with the persistent seed banks and obligate seeding. You can see how it's moving evolutionarily through this lineage of plants. So if you focused only on Arctostaphylos, it wouldn't be as obvious that there's something going on in this lineage that is differentially adapting to the Mediterranean climate and the changes in soils and rainfall patterns and fire patterns. So let's take a look at it from an ecological perspective. The succulent fruit are dispersed by birds, but rodents are involved once you get to dry fruit. They're undoubtedly what's causing the endocarps to be thickened and fused together. And rodents are very interested in summer fruit maturation. On the other hand, the dry fruit and the earlier maturation was probably due to aridity increasing in the summer, because the Miocene had some rainfall in the summer, but every million years afterwards, it got increasingly drier until we got to a Mediterranean climate. Meanwhile, fires are also selecting for a number of these characters. So this lineage is not responding to simple things. And we're only talking about reproductive characters here, but we've got birds and rodents, climate and fires, all being very important and involving the diversity that you're seeing in this group. But meanwhile, Arctostaphylos takes off. And it's mostly due to obligate seeding. And that obligate seeding, as you could see from the Arbutoideae, came from rodents burying seed. And in the context of fire, that selected for open habitats, and the seed became dormant, and you ended up with long-term dormant seed banks. The rodents were doing all that nice burial for them. But once you've got long-term dormant seed banks that are responding only to fire, that allowed the selection for obligate seeding. And now you've got lineages of plants that are more responsive to shifts in soils, to shifts in climate, and other kinds of conditions that might be impacting the vegetation. So it's not a simple story. It's a cool, linear, complicated story. It really makes this group very fun. So the Arbutoideae is a small early family, subfamily of the Ericaceae. There's shifts in climate influences that cause changes in life form. The climate drying changes the fire regime. The habitat shifts changes the animal community. So we end up seeing changes in fruit from fleshy to dry and other kinds of shifts in Arctostaphylos. So now we've got that high diversity in Arctostaphylos species. So let's take a look at what that looks like morphologically. You've got diversity in stem characters. Here we're looking at two different taxa, Eastwood's Manzanita and Brittleleaf Manzanita. They both have smooth red bark that you're familiar with for Manzanitas, but these guys have large burls at the base. So these are resprouting species. You burn them down, they just resprout. And you Southern Californians might be interested in the fact that Crustacea down there on the right-hand side is covered with moss after the first rains. It's in a maritime climate and it's pretty foggy in the summer. You might not be as familiar with the fact that a lot of Manzanitas don't have smooth red bark, but have shaggy bark, which might be gray or slightly red. So tomentosa subspecies have that, and Arctostaphylus rudus from the Lompoc area also has the shreddy bark. But they both have burls, so they're again resprouting groups. This is what they look like, really pretty. A lot of them don't have burls. They might have smooth red bark. There are a few that don't have red bark that is smooth, such as the Nissanena, the Eldorado Manzanita we looked at earlier. The obligate cedars without fire are completely dependent upon their seed banks. And if you look only at stems that are close to the end, the stems that are basically 1 year or less, then there's a lot of pubescence characters that you need to take a look at. Glutinosa has that short canescent hair. Sensitiva, on the other hand, has long, stiff hairs that are non-glandular. The greenleaf manzanita, Arctostaphylus patula, has two subspecies. On the left, the typical one, it has small glandular hairs. The glands are usually yellow. So you might see in keys that they're called golden glands. But there is a subspecies that occurs in Northern California called Gankiniae, named after Roman Gankin. And it has short, often canescent hair, no glands at all. Really very different looking. A lot of variability in leaves. They're usually described well enough in keys for you to figure it out, but every plant has some variation. The two panels on the right are the ones I want you to pay attention to, because Manzanitas mostly have leaves where each side of the leaf looks just like the other side. And they have stomata on both sides as well. So stomata are holes in the leaf where they're able to pick up carbon dioxide and use that in photosynthesis for their energy. The two on the right, though, are different. They only have stomata on the lower surface of the leaf, and the leaves differ in color or they differ in pubescence. The one on the top is Arctostaphylus pajaroensis. The one on the bottom is Arctostaphylus uva ursi. So we call those bifacial leaves. And if you were to look with a hand lens, you would see that the stomata are only on the lower surface. So I know you guys have great vision, and this is what you're looking for. Of course, this is under a microscope. This is what the pores look like. In Arctostaphylos, they have waxy coverings, but there are two guard cells that fluctuate to either open or close those pores. And they close when it's stressful or at night to prevent the loss of water. This is what you'll actually see under a hand lens, lots of little white dots. So the way I do this is you curl the leaf and you hold it over to the sun and you look for those white dots. If you've got a hairy leaf, don't bother. You won't be able to tell. There's also diversity in the inflorescences. So these all have multi-branched panicles. The two on the left have small scaly bracts that are dry. On the right, tomentosa has those big, broad, leafy bracts. And those are important characters for some species. A lot of species only have racemes where there's just a single main branch that the flowers come off of. You have to be careful with these because sometimes those racemes have a small second branch, but they can be scaly bracted like Franciscana on the left, or they can be leafy bracted like Arcanescens in the middle. A lot of diversity in fruits that we've already talked about. There's that Gabelonensis on the left that I said I would give you a close-up of. Really large, big berry Manzanita-like fruits, except they're not sticky on the outside. Or they could be small and depressed globose or apple-shaped like in Nevadensis. And of course, you have to break them open and check out whether the seeds are fused or not. That sometimes is an important character in the keys trying to work these groups up. So let's go take a walk and see how we're going to identify things. So I was living in Pacifica for 30 years, and San Pedro Valley has a county park called San Pedro Valley County Park. And it has two manzanitas. One of them is a brittle leaf manzanita, Arctostaphylos crustacea. We've already talked about the fact that it has a burl. And it also has a local endemic, the Montera Mountain Manzanita, Arctostaphylos Monterences. So I only showed you the stem of this one earlier, gigantic tree-sized plant. But this is really a lovely shrub as well. So how would we tell these two apart if you're trying to identify them? Well, the Montera Mountain Manzanita has smooth red bark, but so does Canescens. They all have white flowers, so they're not going to be a help. But if you look on the ground, Monterences does not have a burl. Crustacea has really obvious burls. So that's an important character. And if you look at the leaves, there's huge differences. Monterenses has clasping heart-shaped leaves that we call auriculate. They have very short petioles. They have stomata on both sides of the leaves, and they have long, stiff, and glandular hairs. Crustacea, on the other hand, has much longer petioles, and the bottom of the leaves are flat and to elliptic, and I just mean the edge. But the surface of the bottom of the leaf only has stomata. The top of the leaf does not have stomata. And sometimes there's a little bit of pubescence on the bottom of the leaf, but not on the top. It also has long, stiff hairs, but there are no glands. So there's really a lot of differences between these two once you're familiar with what the important characters are for this group. Another thing, there are traits that are not always present. So these are photographs taken on late December last year. And Monterensis was in bloom, Crustacea, the brittle leaf manzanita, the buds aren't even swelling yet. So these two plants flower at very different times. And it turns out that there's a lot of plants that are that way, but let's think about the fact that traits that are important are not always visible. So another diversity that you can't see that's very significant in this group is that species differ in the number of chromosomes. Don't you love that? Diploids have one set of chromosomes from each parent. So Archeocephalus patula, for example, is a diploid. Tetraploids, on the other hand, have two sets of chromosomes from each parent. So they have four sets of chromosomes altogether, and that's why they're called tetraploids. What that means is that tetraploids and diploids are not very good at exchanging genes, so they can't really hybridize with each other because they don't have the right number of chromosomes for each other. So whenever pollen meets an ovule, they have problems. And there's a barrier there, but you don't see that barrier. It's not morphological. Another thing you don't see is that this group is old enough that it has two separate lineages that are somewhat genetically distinct. We're just call them a small clade and a large clade from the number of species that are found in each one. And what that means is that they can grow together in habitats and not really hybridize. And so they stay the same. So here's an example from Mount George in Napa County with Arctostaphylos canescens from the large clade and Arctostaphylos stanfordiana from the small clade. You would be able to tell a hybrid between these two because there's so many differences between them, but there's not a single hybrid in the whole region between these two species. Another location with species from two different lineages. Another thing that's cool about that is just like with the Monterences and the Crustacea, those are differences in ploidy levels in that case, but species from different lineages also flower at different times. So Pajaroensis and Hookeri grow together in the Pajaro Hills near Fort Ord. And Pajaroensis tends to flower in January, late December, but Hookeri holds off until February and March. And what that means is there's very little overlap between these two, which means they can't even exchange genes from pollinators moving pollen from one plant to the other. So that becomes a phenological or timing barrier to keep them as separate species. So everyone always thinks that Manzanitas hybridize like crazy. But in fact, if you've got two clades together, there's very little hybridization, and in some cases, no hybridization at all. But if you've got a soil interface where two different soils come together and a Manzanita has specialized to each soil type, you can get quite a lot of hybridization there. But they have to be from the same clade, the same lineage, so that they're very closely related to one another. What that means is when you're in the field and you see species, if you've only got one species, it doesn't really matter. It could be from either clade, it could be a diploid or a tetraploid. But if you have two species together, they're either a diploid from each clade, or there's a diploid and a tetraploid together. And this is super common throughout California. You almost always can find two species together, and they tend to differ in ploidy level, or they tend to come from different clades. Now, Southern California doesn't have very many of the little clade, but it's a much more common circumstance in Northern California. You do have places where there are three species together, and in that case, there's a diploid from each clade and a tetraploid that coexists with them. So what you can see is even with the huge diversity in this group, there's ways to parse them out and to figure out what's going on. It's kind of cool. So how did we end up with so many of these bloody plants? Well, there's lots of reasons for it, but I'll end with just speculating about hybridization, because you saw that within clades, species have little reproductive barriers. So if they have a climate change, and these guys have been around for 15 million years, so running north and south with climate shifts, especially during ice ages and between ice ages, you can imagine they could run into each other and start exchanging genes. Now, between clades, there's very little viable hybridization possible, but that might be the origin of polyploids, because if you've got a genetically difficult group, then it might be that they just double chromosomes by accident, and all of a sudden they're genetically viable. So let's do a little speculation to end. And you can talk about Arctostaphylus refugioensis, the refugio manzanita in the western Santa Ynez Mountains. This plant is a beautiful auriculate leaved plant. That means it's maritime, but it has gigantic Archeocephalus Glauco-like fruit. And that suggests that it might be of hybrid origin between some old auriculate species and Archeocephalus Glauco. Really close by, a little further west and north, is a subspecies of Purissima, which grows near Lompoc. Purissima does. Now, this one in between Purissima and Refugiensis has also small round fruit where the seeds are often completely fused together. And that suggests that there's been a little introgression from Refugiensis into Purissima. And you've got this intermediate with the small fruit, but round. Purissima has the apple-like fruit with the pressed globose, separate seed. But this one, there's often complete fusion, just like in Refugiensis. So possible that there's been some genetic introgression. And then you have a plant like Gabelonensis with white hairy leaves that are auriculate, but again, gigantic Arctostaphylus Glauco-like fruit, this time without the viscid external structure as well. So what has this Glauco been doing? Running around north and south in this California area and just spreading its genes everywhere. So it's just a big possibility. So the high diversity of Arctostaphylus species, well, we know they're very different from their close relatives in the Arbutoideae. They have different life histories of sprouting and non-sprouting. They're very interconnected with fungi, which I didn't talk about, but also with animals like seed predators and pollinators. They have very different lineages, and they have different ploidy levels, and all of these things are merging together to increase that diversity, especially with hybridization, which is possible among some of them. So that ends up with all of these different stories. We've got a subfamily where you end up with a very unusual lineage, Arctostaphylos. You've got Arctostaphylos with lots of different characters in contrast to the other lineages and likely responsible for all of that diversity that you see. So thanks for listening. That's it.
Thank you so much, Dr. Parker. That was absolutely fascinating. We have a lot of questions, and I think our time is almost up, so I'm a little nervous as to how this is going to go, and so I'm just going to start, okay?
Okay.
First question from Susan is, what distinguishes Xylococcus bicolor from Arctostaphylos?
Well, it only has stomata on the lower surface. It has a very different fruit structure that we talked about a little bit. And the internal aspects of the fruit are pretty different. There are some other minor characters, but it's a very close relative. So you would expect them to overlap to some extent.
Okay. Andy asks, is the Manzanita species diversity supported by nucleic acid phylogenies?
Well, we're just getting to that. Almost all of the genus has been genetically typed for one or two genes, and it's not completely supported. But for doing whole genome genetics, it's being basically supported.
Okay. Linda asks, what about altitude effects, which Manzanita, let's see, what about altitude as an effect of Manzanita growth, maybe?
Well, we do have species that are restricted to high elevation, like the greenleaf manzanita and the pine mat manzanita. We have species that get out of the California floristic province, like Columbiana along the coast, that goes all the way to British Columbia. We have species in Arizona and Utah, but they're pretty much high elevation species. That's a moisture limitation as much as anything. Yeah, they grow all over the place in lots of climates, but yeah, there's lots of climatic limitations, except they're adapted to almost all of them.
Okay. Juvia asks, what are the conditions in which manzanitas lacking trichomes grow? Do you often see hairy and non-hairy manzanitas in sympathy?
You can. There doesn't seem to be any geographic or spatial reason for hairiness versus non-hairiness. It's probably a historic thing, or it just might be random in some of these plants. But for example, where glutinosa is found in the Santa Cruz Mountains, it co-occurs with a shiny leaved sensitiva that has no hair on the leaves. You have canescens on Mount George with Stanfordiana. Canescens is canescence, so hairy all over the place. Stanfordiana, no hair anywhere. So they can co-occur. No good pattern for you.
Okay. Kat asks, are Ian Bush manzanitas and Lewis Edmonds manzanitas good to be planted on a slope?
So I'm a classic academic botanist, and I don't know the horticultural names.
Okay.
Pretty much, you should plant manzanitas that are close to the climate in which they're found. There's some spread that you can do, but a really rare one, Archeocephalus densiflora, has a couple of horticultural varieties that are planted everywhere, and it does great everywhere, but not all of them do like that. A lot of them can't be moved at all from out of their narrow climate range. So you need to ask a horticulturalist who knows better than me.
Okay. Tom asks, the few manzanitas at the Santa Rosa Plateau have leaves that look terrible, which Keeley speculated was due to frequently being in the fog at the top of the marine layer. Yet apparently many other manzanitas love the marine layer. Any idea why these manzanitas are strikingly different in the response to fog? They might be close to the rainbowensis.
Yeah, I'm not familiar with the area that much. I mean, I've pretty much visited every place, but I don't remember all the names. There's a host of reasons why you can have a year or two of bad looking leaves, sometimes from pathogens, sometimes climatic, sometimes from aphids. So I don't want to speculate on that one.
Okay. Henrik asks, are serpentine endemics restricted to serpentine soils because they can't compete outside those soils? Or are they really obligate and actually require serpentine minerals or other soil constituents?
Well, that's a cool question. No one's actually done any studies to make it one way or the other. You can take serpentine endemics and plant them off of serpentine and they do fine, or at least some of them do. There are others, for example, there are some that are restricted to granite, like Gabelonensis. It does not do fine off of granite. So it might be a mineral or an acid-base issue there.
Okay. Fred asks, please comment about the incidence of Phytophthora and its threat to manzanitas and the dangers of moving soil and or even carrying mud in on your boot.
Yeah, that's a huge issue. The Ione manzanita, for example, has had huge diebacks from Phytophthora. There are patches of areas in Sonoma County where there have been diebacks. I've studied mycorrhizae, and I know that manzanitas do better if they have their local mycorrhizae, but you don't want to move soil from one place to another. So you can move mushrooms. Yeah, it's a terrible problem. Do not move soil. And you actually have to be pretty careful with making cuttings these days.
Hmm. Carmen asks, are they poisonous?
Um, the fruit are not poisonous. You can make teas. The exocarp and mesocarp make delicious tea. The leaves, they're kind of woody. I wouldn't want to eat them anyway. But the Ericaceae is famous for bad compounds, so the leaves may likely be toxic, but I'm not sure.
Okay.
Other people do study that.
Okay. Cindy over on Facebook asks, is taxonomy based on physical observed traits or molecular data?
Well, it depends on when you're looking. We didn't have molecular data until really the 70s when people started looking at enzyme differences. Before that, it was strictly based on morphology and other things like phenology and timing of things. Now we're being able to look inside the plants for their genetics and to tell whether morphology is a good indicator or not. And the good news is, for the most part, morphology is correct, at least with respect to genetics. But what we're finding is that species are related to things we didn't think they were related to. So that's the cool thing that's happening.
Yeah, that's really cool. Marie asks, why do some manzanitas get a fungus causing branch dieback?
Oh boy, that's usually due to stress. So not a single year's bad drought, but you throw in 3 years of drought or 4 years of drought, and all of a sudden the plants are now susceptible to organisms that have been living on them and in them the whole time. Normally they're able to resist it, but you stress them too much and they cease being able to prevent it.
Okay. Tom asks, to check whether the seeds are fused or not, do you need to try to split the stone with fingers or a tool like a hammer? Or does it count only if they fall apart naturally when you remove the outer part of the fruit?
All you got to do is squeeze the fruit and try to separate the outer part of the fruit from the endocarps. If they're fused together, they'll stay together. If you take a hammer to it, some of them will split apart that would otherwise look fused, but you really have to take a strong hammer to smash big berry manzanita, separate those things. So it's simple, just your fingers.
Okay. Craig asks, how long-term is the long-term obligate seed bank? There is some evidence that large chaparral fires were naturally in the 90-year range.
Yeah, it's likely that the long-term seed banks can last. We don't really know, but probably well over 100 years. The trouble is we don't know what the turnover is in the seed bank. So are any of the seeds in the soil from the first couple of years, or do the rodents eventually find them and it's all newer seed? But there are places where forests have invaded chaparral and covered them up, and there's no more seed input, and the seed has lasted at least another 30 to 50 years or more. For example, there was a fire near Truckee in 1960 that was forest. And the forest completely burned down for the most part, and a stand of Arctostaphylos patula came up. So that stand was at least 60 to 80 years old at the time. So that they do last a long time, basically.
Okay. Julie's asking, what species are symbiotic with Manzanita?
Lots and lots of fungi. They're ectomycorrhizal, but they have a very special kind of mycorrhizae called Arbutoideae, named after the subfamily. It's morphologically a little different, but the species are the same that you would find on pines and oaks. There's great overlap in what they can share with, especially with conifers. In terms of animals, all the rodents are seed predators, but they almost all bury seed to some extent. Even wood rats we've caught on camera burying some seed. Some of them don't bury very much, like small deer mice, Peromyscus maniculatus don't bury very much, but other Peromyscus bury a lot of seed. And of course, all the pollinators, which are mostly native bees, are very important. There was a study out of Riverside where it's not only bees, but butterflies, moths, hoverflies, all sorts of animals utilize the flowers. And if there's a little hint of pink, hummingbirds will go by, use them.
Yeah. Gregory asks, is there a maximum known time that a seed will remain viable during dormancy?
Nobody knows,
Okay.
but probably a long time.
Okay. Craig's asking, any thoughts about why Xylococcus bicolor would have short-lived or transient seeds?
It just hasn't been selected to be different.
Okay.
So that means that physiologically, its seedlings have to be able to establish with a little bit of shade. Manzanitas can't tolerate shade at all, for example. But if you're going to establish seedlings in a non-burned habitat, physiologically, you have to be adjusted to be able to tolerate those conditions.
Okay. Craig is asking, seems like movement along the San Andreas transform fault might have played a role in the hybridization.
Likely climate made it a little faster than a couple of centimeters a year, but the Bay Area does have soil from the transverse ranges of Southern California, but that would have taken a long time. Coyotes and bears will have moved the fruit a lot faster.
Okay. Sandra asked that too. Is it true that grizzly bears played a key role in Manzanita reproduction, or is that an urban legend?
It's not a legend. You can go into the mountains and find bear poop that was just filled with Manzanita fruit. So it's usually in a big pile, but the cool thing is rodents will go and remove all the seed and go bury it. So the bears, the coyotes, foxes, things like that, move them long distance, but rodents do all the burying. And so rodents are actually pretty critical mutualists.
Wow.
I know. You have to like rodents now.
Ron asks, much genetic work has been done on Delmar Manzanita and its relatives. What is the future populations of Delmar Manzanitas that are genetically distinct and not considered Delmar Manzanita?
So that's not an ecology question, that's a policy question, and they're mostly restricted to preserves as it is, but they're mostly surrounded by people. So it's not likely that there's going to be a fire to burn them up. Unless you've got some 10-year-olds and their dogs running through habitats during firecracker season. It's not really clear. The future is up to people conserving them.
Okay. And a follow-up, Ron's asking, will they become a new species?
No.
No, okay. And one final comment, Vince says that you can make tea from the shredded bark.
Ooh.
We have tons and tons of comments and a lot of LOLs and smiley faces for your jokes. So yes, they were well received. Christine even asked, was that shale joke tongue in cheek? Ha ha ha. And then a lot of people just appreciating all your time and effort and for a wonderful presentation. So thank you so much. Really wonderful to get all this information. Got us all really excited about Manzanitas. And we'll pardon you about your shade that you put on the rams also this time.
All right, it was a pleasure. So even though I can't see everybody, I'd like to be able to interact more directly, but I'm always happy to talk about these guys.
Excellent, thank you. And thank you to all of you joining us in on the presentation. Remember, it'll be available on our YouTube channel and our Facebook page. And visit our website for all the information that you need, and that is cnpssd.org. Good night.